Hi, and thanks for the opportunity to talk about my work. I'd like to thank FAIR and IARS for their continued support throughout my early career development. I was supported by a research fellowship grant in residency, and this provided key preliminary data for the IARS Mentored Research Award, which I'll discuss today. And all of this has been foundational to my continued move towards research independence. My focus has been the translational neuroscience of endogenous analgesia. We know that the brain and spinal cord strongly modulate incoming nociceptive input through opioidergic and non-opioidergic pathways. Placebo analgesia also works through these pathways and can be quite powerful, equipotent to 5 milligrams of IV morphine. The problem has been how to leverage this endogenous system for pain management without side effects. Translating placebo analgesia may address this problem. So what's known about it and what are the barriers? There's evidence that expectations about pain relief, or expectancy, is key in placebo analgesia. This is often studied with the experimental scheme shown, where participants are told to expect analgesia, and then the experimenter surreptitiously turns down a painful stimulus to reinforce that expectation during a training phase. A major barrier to applying this paradigm, obviously, is deception, which would be unethical. There's also evidence that associative learning is key to placebo analgesia. This comes from less studied paradigms, which involve the pairing of an analgesic drug with a cue until the cue causes analgesic effects alone. This is challenging to do in the lab for several reasons. To address these barriers, I developed a novel behavioral paradigm of conditioned analgesia using a psychophysical phenomena known as offset analgesia instead of the drug. This might allow a tractable way to study associative learning and potentially translate it to pain management. So what is offset analgesia? Offset analgesia is the disproportionate decrease in pain intensity with small decreases in noxious stimuli. It's shown here, and this is an example from one participant, where that drop in the heat stimulus on the forearm, shown with that arrow, is accompanied by a big drop in pain intensity that's rated continuously on a computerized visual analog scale. And this is different from adaptation by a comparison with a constant control stimulus, shown in the orange dashed line. And we can use offset analgesia, shown with blue, as our unconditioned response in a classical conditioning paradigm, essentially pairing cues that we're interested in prior to the drop in pain intensity. So this is what we set out to do. Initially, we used a visual cue that is the blue screen with the words pain relief on it. This was paired 9 times with offset analgesia in a conditioning phase. And you can see in the middle panel the mean pain intensity over time of a group of participants at that first trial of the training phase. And you can see that that pain intensity drops with offset analgesia, as we would expect, and the participants are looking at the visual cue. Then in the test phase, there's no temperature decrease in the thermode, and this would reveal any analgesic effects of the cue alone. And you can see here that there's a pretty big decrease in pain intensity when the cue is presented, despite there being no change at all in the noxious heat stimulus. This is distinct from distraction because there's no effect on pain intensity of a novel stimulus. Additionally, it's distinct from adaptation because there's no drop in pain intensity without any cue being presented, as shown in that bottom panel. As part of a larger study, participants were randomly assigned to be in a visual cue group, an auditory cue group, or an audiovisual group. And this study demonstrated that contingent pairing of visual or audiovisual cues elicited cue-specific analgesia in the test phase. On the other hand, simple auditory tones and the non-contingent control group did not show conditioned analgesia. Overall, this is a start for a few next steps. A translational application of this paradigm is to combine the visual conditioning cue with IV-PCA analgesic delivery, which may decrease opioid consumption. Before we do that, I plan on using this paradigm to clarify extinction rates of cue-induced analgesia, clarify expectancy effects, and also optimize cue delivery. In related projects, I'm looking at neurophysiologic correlates of conditioned offset analgesia with ambulatory brain imaging and autonomic measures. Thanks for your attention.